be given by Rafael Yuste. He is professor of biological sciences and neuroscience at Columbia University. And he'll be talking about simultaneous imaging of neural activity in 3D.
I'm delighted to be here. I was originally trained as an MD, and the purpose of medicine, the goal is to help mankind by curing diseases. One of the major challenges that medicine has are brain diseases. So my cousin is schizophrenic. He was diagnosed when he was 18, and there's no hope for him. And just like my cousin, many people you know, in your family, your friends, Alzheimer, schizophrenia, autism, many forms of epilepsy, many forms of Parkinson. It's essentially the dark corner of medicine where we cannot do anything about these patients. It affects a large proportion of mankind, and the reason is because we don't understand how the brain works. We cannot fix the problem if we don't understand how the machine works. Why don't we understand how the brain works? People have been looking and studying the brain for more than 100 years, generation after generation. Some of the smartest people of each generation devoted their lives to this. What's the problem? It turns out that over the last century, most of what we've learned about the brain has come from the studies using electrodes, in which you record the activity of individual neurons in an animal or in a human, and then you try to draw conclusions about what does the brain do based on that data. No? The problem is, as you can see in the bottom left here, is that any piece of the brain you look at, in this case the cerebral cortex, is composed of hundreds of thousands of neurons that are interconnected in complicated networks. So to try to understand a system like this by looking at individual neurons is a little bit like trying to watch a movie on a TV if you had to look at single pixels. You will never get it, because the picture of the TV is an emergent property of the correlation between all the pixels. Now, there's been some techniques like fMRI that try to look at the activity of the whole brain, but they don't have the resolution to show us what single neurons do. So I would argue that the problem is essentially a technical problem. We need to be able to monitor the activity of neural circuits, seeing what single neurons do, but seeing the whole picture of the TV. So how do you do that? So you can try to do this with electrodes, but you can quickly come to the conclusion that it's a dead end. You're not going to be able to put electrodes in every single one of these neurons that illustrated here. Every white dot is a neuron. So the reason I'm here is because maybe the solution lies in optics and using light to try to take those measurements. And this is a piece of a line of work that started when I was a grad student in the lab of Larry Katz, who was deceased recently. And we discovered that you could use calcium fluorescent indicators to monitor the activity of neurons due to the fact that whenever a neuron fires, as illustrated here, the action potential of the neuron lead in an influx of calcium into the cell, because every single cell in the body has these calcium channels on the membrane, and there is an unequivocal relation between firing of the neuron and calcium influx. That means that if you can monitor the calcium, as you can do here, you can essentially track the activity of entire populations of neurons. So in this case, every gray dot is a neuron. This is 10 minutes in the life of the cerebral cortex of a mouse. Something like this is probably happening in your cerebral cortex right now. And whenever the neuron turns red, it's because the neuron is activated. And you can see there are patterns of firing which are quite mysterious, and they must carry the substrates of our thoughts, of our minds, and when they're disorganized, or these brain diseases. So that was part of the toolbox, but another big part came from the use of nonlinear microscopy to photomicroscopy, as you've heard from Peter. So, this was work done in collaboration with Winfried Denk when we were at Bell Labs, and we applied to photomicroscopy to image deep into nervous tissue. Here are little pieces of a neuron, dendrites of pyramidal cells in the cortex when they're actually, we found they're moving around, and that again is probably happening in your brains right now. These neurons are not static, they're sort of moving and changing their shapes. So now you can do this type of measurements in living animals. In this case are now data, more recent data, of an awake behaving mouse. The mouse is running on this treadmill, is looking at this screen, the skull is open, and we have a two-photon laser coming from the top, and we're taking these images, these movies that are shown on the bottom left. This is the activity of this group of neurons in the visual cortex. This is the part of the brain that deals with understanding the image that we're showing to the animal. And the bottom right is the analyzed version of this movie. This is the raw data, and this is the analyzed version. So these patterns of activity are showing us that these neurons are not firing individually, they're forming groups. So there's something truthful in this idea that there could be an image here, like a TV image, built by the correlation between neurons. Now, in order to try to do this, you have to do this in 3 dimensions. And the problem with fluorescent microscopy, with laser microscopy in general, is that if you scan the image, if you want to image things that are in different focal planes, you're going to have to do this seriously. So we introduce into the field the use of spatial light modulators in order to build a hologram of light, because we can actually arbitrarily shape the light into any 3D structure. And we solve this 3D problem by simultaneously imaging. Instead of rastering every cell, we don't raster anymore. We scan, we sort of do a scanless type of microscopy, and we generate little beamlets of light, each of them targeting the particular neuron that we want to image. So the experiment, the typical experiments, we first find out where the cells are, and that we do with conventional slow raster two-photon microscopy. And then with an algorithm, we quickly find the position of every cell, the optical center of mass, and then we upload these coordinates into the SLM that splits the laser beam into a bunch of beamlets, each of them targeting a cell, a neuron. And then we use a camera to simultaneously gather the fluorescence from all these cells. That works, but we had to deal with the problem that microscopes, since the time of Leeuwenhoek, have been designed to image things in 2 dimensions. So we needed to aberrate the point spread function of the microscope, and we did that using a cubic phase, a little quartz phase mask that we put in the emission path of the microscope, and we turned the point spread function from a little football into sort of like a pencil that penetrates the whole sample. And this enabled us, you can see in this demonstration, to image an object. In this case, it's just a test pattern going through just media. And there's one test pattern which remains in focus, and you can see with a conventional two-photon microscope and with our 3D SLM microscope. But there's a second pattern that goes up and down a millimeter through this tissue phantom, and you can appreciate how you can actually still image this other pattern, even though it's completely out of focus for the conventional microscope. So this is using this face mask PSF design. So using this method now, we can image, I would say, for the first time, the activity of neurons in 3 dimensions in entire brains. And this is now an example of the brain of a larval zebrafish, which is actually very convenient because it's transparent. And here you have the simultaneous measurements of 50 neurons. This is just an example of a test. And they're labeled with these calcium indicators, and they're firing spontaneously. And you can see in XY, XC, or YC how the firing pattern occurs in the brain of this animal. So our hope is to take this technology and little by little push it into the clinic, and push it into maybe miniaturize these microscopes. This is on the left. This is our original SLM two-photon microscope. Here on B, this is a smaller version of an SLM microscope that we built in collaboration with BNS, Boulder Nonlinear systems. And the hope is to be able to take advantage of the fact that the spatial light modulators are universal optics, and try to make miniaturized microscope that we can in the future maybe put on a human, and be able to take these movies like the ones that I showed you from a mouse, except from a human patient, and maybe use that data to try to understand a little bit better what's the problem with these brain diseases. Now, you may think this is completely crazy, and many of my colleagues told me, you guys are completely out there, but we were persistent, and we actually wrote up these ideas and were able to convince the White House. And this was the original of the BRAIN initiative. So our original proposal was called the BRAIN Activity Map, and the goal was to build a systematic technology development effort across many labs, across different countries, mimicking the Human Genome Project, in order to develop methods to monitor the activity of every neuron in a piece of brain, either in the entire brain of a small animal or in a piece of brain of a human, and that was our first goal. The second goal was to manipulate, to change and alter the activity of these neural circuits in a way that's systematic. And the third goal was to computationally model these circuits so that we could understand what they do. So the White House sponsored the idea. It's become the BRAIN Initiative, and I would say the good news for people like you that are in a way tool builders is that there's at least the recognition of the scientific and clinical communities that maybe the future of some of these devastating diseases lies on the hands of people that are actually building the methods to try to diagnose them a little bit better, as we've heard, or maybe come up with ways to understand what is the pathophysiology so that they can lead to better therapeutics. So I leave you with this quote of Sidney Brenner, that maybe the untold heroes in the story are the people that are actually methods builders. Thank you a lot.
Thank you very much.